I feel very fortunate to be part of this ViroiDoc project. I think this is fantastic, and I feel honored to give this lecture about taking a peek at viroids. I guess this is a problem of age, actually, because I started to work with viroids back in 1991 when I started my PhD. Okay, so I guess in the audience, I know that in the audience there are we have big viroid experts, that's for sure, but possibly we have also people that are not that familiar with viroids. So particularly for those that are not that familiar, viroids are relatively small RNAs. You can see in the lower part of this image, you can see the sequence and a kind of outdated secondary structure of potato spindle tuber viroid, which is like the prototype viroid. You see here that it's a single-stranded RNA, circular RNA, 359 nucleotides. That's why, because I said it's relatively small, because this was considered small some years ago, but we know a lot now about very important RNAs in the cell that are way smaller than this. You see here that viroids have a very particular structure. It's a very compact structure, highly base paired, but with some particular properties that you can clearly spot from this image. It contains some double-stranded regions that are not that long, interrupted with loops and bulges. And all viruses are this way, and possibly this is an important feature of viral molecules. Another important question is that, to our knowledge, viroids are non-coding RNAs. Nobody has been able to demonstrate so far a protein product that is produced from a viroid. However, this is definitely an open question, because maybe in the future we will be able to detect small peptides coming from here or there in the viroid molecules. Also very important, there are viroid-like RNAs that encode proteins. So this is definitely something that we shouldn't just give like a definitive answer that viruses are non-coding RNAs. Okay, one thing that is that we can say for sure is that viruses are infectious agents, and this is one of the reasons why we are here together, because they induce disease in higher plants, and in some cases these diseases are important, and this is something that we have to take into consideration for crop protection and for food security. During these last decades, since we have been doing research in viroids, we have defined a series of viroid species. This is again an outdated classification. There are a few more species now that are accepted. One very important issue here in the viroid classification is that apparently, or more than apparently, to our knowledge, we can differentiate two very different types of viroids. Those that are gathered in the family Viroidae have some particular properties in the molecule, and very important, they accumulate and replicate in the nucleus of infected cells. In contrast, some other viroids that are gathered in the family Viroidae replicate and accumulate in the chloroplast of infected cells. These viroids, one of the interesting properties is that they contain hammerhead ribozymes. Either in the Viroidae or post-Viroidae family, viroids replicate through an RNA-based rolling circle mechanism, no DNA intermediates. Actually, this is what I started to one of the topic of my PhD. Actually, I started to work with this back in 1991. Although by those years we already knew a lot about this replication process, but well, I contributed to with some refining some steps and also with the host factors involved in replication. So rolling circle mechanism, I don't want to get into details because this could be a little bit complex, particularly for those that are new in the field. But we have circular templates that are recognized by RNA polymerases that produce complementary strands. Then these strands can be processed or not. In the case of the nuclear viroids, they use host RNases. In the case of chloroplastic viroids, they contain their own hammerhead ribozymes. At the end, during replication, monomeric linear viroids are produced that are circularized to produce the progeny, the circular plus strand RNA. As you can see here in this image, I wrote the names of some of the host enzymes involved in replication. We have a very interesting issue here is that some of them are DNA-based, no, not DNA-based, but enzymes that in the cell are involved in DNA metabolism. For example, the RNA polymerase II or the chloroplastic RNA polymerase, both of them use in the cell DNA templates. However, related to virus replication, they are able to copy an RNA template. The same can be said with DNA ligase 1, that, as the name indicates, in the cell acts as a DNA ligase. However, the viroids manage to recruit this enzyme to ligate the RNA monomeric intermediate. Okay, so as I mentioned before, viroids, the discovery of viroids and most of the history of viroid research is associated to higher plants. Okay, the first one, potato spindle tuber viroid. I will talk a little bit more about this in a moment. Citrus exocortis viroid and some other viroids that, as you can see here, produce symptoms in different crops: cucumber, chrysanthemum, avocado, peach, almond. And this is one of the reasons why viroids were studied, because this disease was very important in agriculture. I'd like to mention here that I said at the beginning that for sure viruses are infectious agents. We have to be a little bit careful when we talk about viruses as pathogens, because there are some viruses that do not produce any symptoms in the infected plants. This is a good example. For example, is eggplant latent viroid. Eggplant latent viroid is an infectious agent that can infect eggplants. However, we are not able to see any symptoms, any difference between the infected and the mock inoculated plants. Although this is something that is difficult, again, because sometimes these differences could be at the molecular level and we cannot appreciate. Let me show you here another example. Pitch latent mosaic viroid is a viroid that, as the name indicates, was considered latent at the beginning. However, an insertion of this little sequence in blue in one of the parts of the molecule changes the asymptomatic variant into a very symptomatic variant that it produces this Y phenotype called calico. So the frontier between pathogenic and not pathogenic is kind of diffuse sometimes. All right, so a little bit of history. There is little discussion that the father of viroids is this man here in this picture, Theodor Diener. Theodor Diener was, he passed away very recently. He was a fine plant pathologist born in Switzerland, but he was working in the USA. At the end of the 60s, at the beginning of the 70s, he was publishing an amazing amount or a good amount of amazing papers that I invite you to read because they are really inspiring and definitely amazing. You have to take into consideration the techniques that they were using back in those days. So Diener was studying this disease, the spindle tuber disease of potatoes. I'm showing you here a paper in Science, 1923. It's it's extremely short, just one column. I invite you to read because these authors, Schulz and Folsom, in 1923, were describing a series of experiments, very summarized, in which they clearly indicated that this was an infectious disease. So the disease was quite an important disease because it affected a very important crop, potatoes, and Diener was studying this, as I have said, late 60s, early 70s, and he found that the properties of the causal agent of the disease were really completely unexpected and very different to viruses. And in a paper in 1971, he coined the term viroid because at that point, he was convinced that a regular virus was not involved in the transmission of this disease. Of course, Diener was not the only one. Many other people back in those days and in the next days were also working with viroids and made amazing contributions. I love this particular paper published by Diener and Reimer. Apparently, Reimer was working with this disease even before Diener. And in this paper, published in Science 1967, they showed already most of the properties that are known now for viruses. And this is a publication by Diener, 1972, which I consider a remarkable figure for the viroid history, because he showed that band number 2, or peak number 2, this was a preparative electrophoresis separation. Peak number 2 contained the PSTBD infectivity, and when he analyzed the nucleic acid that was in this fraction, he realized that this was an RNA. All right, more important contributions. Heinz Ludwig Sanger, working in Germany, his group showed that viruses were circular because they couldn't label they couldn't find a 5' end. And very important, this is also an absolutely remarkable result that was published in 1978. They were able to fully complete the sequence of PSTBD. I have worked with the same isolate for many years, and I have resequenced this isolate over and over again, and I can tell that they didn't fail a single nucleotide. So an important milestone in viroid history. Robert Simons showed that some viroids contain Hagerhead ribozymes. And I also would like to mention Ricardo Flores, because he was my PhD director and also was the director of some of the people that are involved in this network, and also he had a big influence in the young, or maybe now not that young, viroid researchers, particularly here in Europe. I just finished my talk saying that starting 2023, the most of or an important paradigm, I will say, in viral biology started to change, because there were two very nice publications showing that viroid-like RNAs can also apparently replicate in fungi. Are they viroids? Should we call them just viroid-like RNAs or mycoviroids? I don't know. Thank you very much for your attention.